Hello, Assalamualaikum. This video is an introduction to the cell division chapter 4. So to start, we are going to learn the key roles or importance of cell division. Cell division is for the continuity of life, whereby all organisms in earth, they need to continue their life or for the generation of their existence. Cells division is to produce more of their own kind. Cell division is used to distinguish living things from non-living things. For unicellular organisms, cell division reproduces entire organisms, thus increasing their populations. For sexually reproducing organisms, cell division is used to develop from a single cell to fertilized egg or zygote, and most of the cell division is used for renewal and repairing tissue that damage. So these are the examples of organisms that use cell division. For example, unicellular organisms like amoeba, they use cell division for reproduction. For multicellular organisms, it depends on cell division for development from fertilized egg, for growth or for repairing. There are different of types cell division between prokaryotic and eukaryotic cell. For prokaryotic, for example, bacteria and archaea, they use cell division, what we call it as binary fission. For example, in this diagram, one cell producing two genetically identical daughter cells. Cell division is more complicated for eukaryotic organism because they have multiple chromosomes and multiple organelles. And eukaryotic uses mitosis and meiosis depending on the type of organisms they are. For unicellular organisms, they use cell division for asexual reproduction, for example, amoeba. For multicellular organisms, they use mitosis for growth and tissue repairing. And other than that, eukaryotic also use meiosis for sexually reproduction. They produce gametes, whereby they produce egg and sperm. During cell division, the cells must copy the genetic materials, aka the DNA, and all the DNA in a cell constitute the cell's genome. In prokaryote, the genome is often in a single circular DNA molecule like this diagram. And for eukaryotes, the genome consists of number of DNA molecules. For example, human genome consists of 46 chromosomes. So in this video, we are going to focus more on cell division in eukaryotic multicellular organisms. So in eukaryotes, the genetic materials that need to be passed to the daughter cells are located in the nucleus. Human genome consists of 3 billion base pair of DNA. If you look at the cells of DNA under microscope, you can see that DNA is actually organized into structure called chromosomes. So in this diagram, the eukaryotic cells, in this diagram of eukaryotic cells, the chromosomes are enclosed in the nucleus like this diagram. Eukaryotic chromosome consists of chromatin. So chromatin is a thread-like structure. The chromatin consists of a DNA as well as the proteins that condense during cell division. During cell division, the chromosomes, the chromatin must be condensed. How it condensed, it coiled up into tightly packed and shorter form so that it can be easily separated during cell division. And the eukaryotic genome can be either haploid or diploid. It is the number, haploid half, diploid is the double. So in human genome, somatic cells, for example, non-productive cell, it has a total 46 chromosomes. We call it as diploid. While for human gametes, it has half of chromosome as somatic cells, which is 23 chromosomes. We call it as haploid. So this is how chromosomes look like after being replicated during cell division, from a single chromosome to double chromosome. We do not call it as double chromosome, we call it as one chromosome. So one chromosome consists of two sister chromatids. So there are few terms that you need to know for cell division. For example, sister chromatid. So sister chromatid can be defined as an exact copy of the other, and each contains one DNA molecule. And then there's centromere. So centromere is the point where two chromatids bound together. And this the P arm and Q arm, and this the DNA molecule that coil up and tightly packed into a shorter form of chromosomes. So this one is the level of DNA packaging in eukaryotes, whereby chromosome consists of tightly packed chromatin. So chromatin consists of DNA also, and also the histone. Histone is the protein, and then the nucleosome dedicated to the terms that wrap DNA by the protein histones.